This talk is going to be, well, hopefully a nice, we're getting towards the home straight now, and I'm hoping that this will be a good, almost a synthesis of some of the things we've already heard this afternoon. Particularly, I mean, Eric's talk just now has encapsulated beautifully about 90% of the problems that I have. So, so what I'm going to do here is, first of all, tell you a little bit about the organization, if the button works. Right. So the Wellcome Trust Sanger Institute, that is our lovely campus, we're part of it. Potted history. We were founded in 1993. We were part of the public project to sequence the human genome. Our original remit was to sequence 1/6 of the human genome in that project. In fact, we ended up doing slightly more than a third of it. We were the single biggest contributor to that project. And you can see in this timeline that we did a few little things along the way. So, opened in 1993. 1998, we completed the nematode genome, which was our sort of practice run. Pick a small organism, have a go. The first draft human genome came out in 2000, and in 2003, at least to some definition of completed, we said we'd finished the human genome. And that was also the year that we sequenced our 2 billionth base pair. And that's worth remembering. It took us 10 years to sequence 2 billion base pairs. After that, we started diversifying our scientific program somewhat. Still mostly based around DNA sequencing. But we had a big step change that came along in 2008, which was when, as any of you who were involved in next generation sequencing, that's when next generation sequencing came along, which gave us our big headache, which is still continuing. So up until that point, IT was getting cheaper faster than sequencing was getting cheaper. So everything was nice. That is no longer the case and hasn't been the case since 2008. When we bought our first next-generation sequencers, we immediately replaced all of our current sequencing capacity with 2 machines. We used to have 100 capillary sequencers. We replaced them with 2 next-generation sequencers, and that was the same capacity. In fact, we didn't replace them with 2. Of course, they went and bought 20. And they did it pretty much without warning IT either. So we had an interesting couple of months in late 2008 where we suddenly had to throw together a lot of kit. We've been joining various international consortia involved in large-scale sequencing projects. So you can see this one in 2009. We joined the International Cancer Genome Consortium. Now that's a really good example of a problem where we're going to have the same problem that Eric was talking about, because we all want to access each other's data. How on earth do we do it? And the other thing to note about this slide is that as new sequencing projects came along, they seem to get bigger by about a factor of 10 every 2 years. So that's, again, considerably more than Moore's Law. So we had the 1000 genome project that started in 2008. It completed a couple of years later. In 2010, we started the UK 10K project, which is 10,000 genomes. That has just finished. And as some of you are aware, Sanger are not necessarily involved in this at this point, but NHS England have just announced that they're going to sequence 100,000 genomes. So this is just going on and on and on, and it's just going to get worse. It's quite a pleasant challenge to have. So we're funded by the Wellcome Trust, the world's second biggest medical research charity. And I've already said that. We have, at the moment, around 17,000 cores of compute in total. And we have, at the last time we counted it, 22 petabytes of usable storage. The scientific program, it's worth describing this a little bit because it gives some insight into just where our problems come from. It's currently divided into 5 broad programs. We have pathogen genetics. So one of the Wellcome Trust's primary aims is to look at disease, particularly that affects parts of the world where and conditions that the pharmaceutical industry is not generally terribly interested in dealing with. So we have a lot of collaborations with researchers in Africa dealing with nasty African parasites of various sorts. And so there's a lot of sequencing of those organisms as well as sequencing patients who've got those diseases. So you're looking at trying to establish whether somebody has a particular form of a susceptibility to a disease, or whether they're going to respond badly to a particular drug treatment, whatever it might be. Human genetics does a very similar sort of thing. We're looking at the sequence of human beings to identify what variations may be responsible for susceptibility to cancer, susceptibility to diabetes, for example, conditions of this sort. We then have, I'm going to do cellular genetics next, actually. So it's hard to say. Cellular genetics is a new facility we have within the organization, where we look at, once you've got a mutation in a cell or a tissue, let's actually record in some detail what that does to the cell, because then we can do interesting statistical comparisons between what's happening to the cells and what the actual genetics were that the sequencing program produced. The mouse and zebrafish genetics research program is the same thing, but the next scale up. Living things are complex and interrelated things, and when you've mutated a gene, it's not really enough to know what it's going to do in a single cell. You actually have to know what it's going to do in the whole organism. So there's a whole program of generating experimental animals, mouse and zebrafish, with specific genes disturbed so that scientists can produce useful models. An example of that we've done that received some note was we have a good mouse model of congenital deafness, for example. Then we have, finally, bioinformatics, looking at the algorithms which are actually involved in doing this analysis. And some of the best results we've had that have reached the press and been useful, just to give you some idea of where this can go. Pathogen genetics. We had a nice, if any of you were at HP Cast, Guy Coates talked about this at his talk there. We had an infection outbreak in the maternity hospital in Cambridge, the Rosie Hospital. And the infection control people wanted to identify the cause. And what we were able to do was take samples from the cases that had occurred, sequence them very quickly, turnaround was less than 48 hours, show that they were all the same strain of bacterium, and therefore they must have come from the same person, because these bacteria tend to mutate quite quickly. So there was a single source of infection, and then they could correlate that with duty rosters and so on and identify the person who was infected who didn't actually have any symptoms and sort out the problem. So supporting all of this, we have a number of core facilities. The model organisms and cellular generation, those are the same things that I was describing earlier. Those are the core facilities. We have DNA pipelines, which is our industrial scale DNA sequencing facility. And then we have, of course, IT, which is underpinning the whole lot. So an ideal data flow that we all have to deal with. Somebody's collecting some data, somebody else wants to analyze that data, somebody else eventually, well hopefully somebody has some good results that they want to publish. And those three things require somewhat different storage architectures. A more concrete example of this is in these sort of variation studies that I've been describing. So the first thing we do is collect the data. So we fetch the data from the instrument, we do some automated quality checking on it, and we align it to a reference genome. After that, the scientist may want to align that with other data that has previously been sequenced. They want to analyze the whole set of data so that they can find out where the variants are, which is the variant calling part there. And then they'll perform some sort of statistical analysis, which I frankly don't understand, with the phenotype, which might be disease state, drug resistance, and so on. And then they want to publish it. So they produce things like, we have a thing called COSMIC, which is the Catalogue of Somatic Mutations in Cancer, I think that stands for, which is a publicly available database describing what we've discovered so far. So we have a lot of virtualized websites and all that sort of thing. So here's what that looks like in terms of a real data flow within our organization. The data comes from the sequencer. We put it onto some staging storage. So that's one storage architecture, whereupon we do this quality control and alignment. We then store that in an archive using iRods, which and as Eric mentioned. So iRods manages that and stores all the metadata to do with the experiment, so that when a scientist wants to retrieve data of a particular kind, it's very easy for them to do it. So this part on the left is a very automated process. On the right, this is the much more sort of free-for-all part of the world, where the scientists will stage data out to our Lustre file system, do some sort of arbitrary analysis, write an enormous amount of data back and hopefully not blow things up too badly. And once they've got their results, they'll put it into archival storage or they'll publish it on the website or whatever. Now, choosing the technology to underpin this, we've all seen this diagram any number of times. Really, we all want all three: capacity, performance, and price. Of course, like most of us, we normally can't afford all three. Scientists like to spend their money on sequencing. They don't particularly like to spend it on IT. So this has always been a challenge for us. But I heard Jeff earlier. He said it was all about price. So let's look at these particular types of storage in a bit more detail. The first thing we do with the staging storage is actually this is really we make this as simple as possible. The performance is not a big issue here. All we want, as long as the performance is enough that we can process the data before the next sequencing run comes along, we're fine. It does not have to be ludicrously fast. So what we actually have settled on is this simple scale-out architecture. Each sequencer has associated with it an NFS server with a bunch of direct attached storage, basically. It's as simple as that. It doesn't have to be complicated. It runs Samba so that the Windows machine that's actually attached to the sequencer can talk to it. And then the 1,000-core cluster that actually does the automated analysis then reads the data over NFS. There's nothing particularly complicated about this. So I think we'll move on, because that's... So the iRods side of it. This is where it gets a bit more interesting. iRods is effectively an object store. How many of you actually either use iRods or are aware of it? Oh, quite a few. Okay, so I don't need to explain what it does. But the great advantage is from our perspective, it's a really nice, rare occurrence of a piece of IT technology that both the system administrators love and the users love it. This is a rare thing, so it's really nice to actually have something that does this. It's vendor agnostic. This is a really big thing. I know this is a single vendor meeting here, but the real world is the real world, and we need to be able to cope with multiple vendors of kit. So it's mostly running on DDN SFA 10K at the moment. It does have a few other vendors' bits of kit in it. It's currently done purely as block storage presented to the iRes server. There's an Oracle RAC cluster holding the metadata. And we have 2 copies of this. The iRods is managing the replication across 2 copies in physically distinct data center quadrants. They're actually in the same building, but there's an 8-hour firewall between the 2. Because it's done as block storage, this is actually done as a series of 43 terabyte LVM volumes from each of these storage servers. That's quite a lot of, given that it's 4 to 5 petabytes of data in total, this is actually quite a lot of LUNs that we're having to manage and file systems. But that's done so that if there is a problem, doing the file system check is not too onerous. But obviously this leads to some ways that we could improve this in the future. Once you get downstream, you're into getting into Lustre world and the much larger number of clusters that we've got available there. So what we do there is that we've got several clusters and several Lustre file systems. The reason for that is twofold. Firstly, most of our code is single-threaded. It's quite metadata heavy. It makes sense that we increase the metadata performance by having multiple file systems. It also allows us to do maintenance without taking everybody out of action, and it also gives us. There was a third reason. I'm beginning to sound like that Monty Python sketch. I can't remember. I'll remember it in a minute. Oh, that's right. So it's the other reason, of course, is that once if there is a problem with any of these things, we deliberately set these things up so the large particular programs have their own set of infrastructure, both Lustre and in some cases their own cluster. So the Cancer Genome Project, for example, has its own. And that means that if anything does go wrong, then the only people, especially if it's done by, as has been said by others earlier today, if it's user error, the only people they've really upset are their own group, which helps a lot in self-policing. So we currently have 11 file systems, I think. Is it 11, Guy? 11, yeah, I've got that right. They're between 500 terabytes and a petabyte each, and they're essentially cookie cut, the same architecture in every case. We're using exascaler hardware, but it is our own Lustre install. We're currently running, because these things have been added over time, a variety of versions from 1.8 up to 2.4. We're in the process of upgrading them all. We don't use InfiniBand except between the OSS and the OSTs. We just have we have 10 gig and some 40 gig networking between the servers and the clients, and we aim to deliver 5 megabytes a second per core of the compute infrastructure. So where are we going from here on? This is pretty much my final slide. You've seen what our science is. You've seen what the sort of challenges that we've got. So here's where we're sort of thinking of going from now on. The first thing is, with iRods, it is an object store. Why on earth wouldn't we put an object store underneath it? So, and then get rid of all of those problems with managing these file systems that are underneath it and all of that stuff. So one of the things we're going to be looking into in the coming year is putting object storage, such as WAS, underneath it. And in particular, we've engaged with DDN in developing and enhancing their current iRods WAS driver, because that's an important thing for us, so that iRods and WAS don't get confused as to who's actually doing the replication here. Is it iRods? Is it WAS? We've also got a challenge coming up around clinical sequencing and personalized medicine. Ultimately, that's what everything that we're doing is about, is getting to the state where we can provide techniques that can be used in hospitals, in doctor surgeries, where we can enable better diagnosis, better drug delivery. In theory, we could, for example, if we've identified a mutation that causes somebody to have an adverse reaction to a particular drug, we can then say, well, there's a diagnostic test which we can do with a sequencer to say, okay, that means that's not the right drug for you. You can go and we can get you something else. Things of that nature. Now, all of that has security implications, which we're not currently set up for. We were originally, as I said, part of the Human Genome Project, and the whole idea was shovel the data out of the door as quickly as possible, and basically to stop the commercial genome companies from patenting it. So our whole infrastructure was not set up with that in mind, so we've got a big challenge around getting that right, and the federated authentication and challenges like that that Eric was talking about are a challenge for us in the future. We also have issues around how we can do this in really exotic places. I'm currently involved in a potential project to try to set up a mini version of what we've got at Sanger in the middle of Uganda. This is going to be quite a challenge, I suspect. It's not going to be on a big scale, but it's going to be tricky. We are going to be moving Lustre, where we're going to be investigating 2.5, again, because we really like the idea of those HSM features that everybody else has mentioned already. So we love the idea of the exascaler sort of commoditized version of Lustre. I mean, I agree that Lustre has got a great deal easier to manage over the years. We've been using it since 1.4 as well. It's a damn sight easier than it used to be, but nevertheless, it would be really nice if it was just something that you go plug in, go, fine, forget about it. And exascaler is a possibility for dealing with that, but at the moment it isn't a current enough version for the features we need. Sequencing technology in the future. We also have issues around how we can do this in really exotic places. I'm currently involved in a potential project to try to set up a mini version of what we've got at Sanger in the middle of Uganda. This is going to be quite a challenge, I suspect. It's not going to be on a big scale, but it's going to be tricky. We are going to be moving Lustre, where we're going to be investigating 2.5, again, because we really like the idea of those HSM features that everybody else has mentioned already. So we love the idea of the exascaler sort of commoditized version of Lustre. I mean, I agree that Lustre has got a great deal easier to manage over the years. We've been using it since 1.4 as well. It's a damn sight easier than it used to be, but nevertheless, it would be really nice if it was just something that you go plug in, go, fine, forget about it. And exascaler is a possibility for dealing with that, but at the moment it isn't a current enough version for the features we need. Sequencing technology is going to bite us pretty soon. We've actually had a bit of a lull for the last year that sequencing has not got any cheaper, which was a bit surprising. We think this is actually a lie, that what's happening is the sequencing manufacturers are sitting on their hands. They've got improvements that they could release, but they're waiting for new technologies like nanopore sequencing to come out, which are going to give us a severe problem, even worse than our current sequencing capacity problems. Especially because some of those sequencing technologies, Oxford Nanopore, for example, are suggesting a sequencer which is effectively a thumb drive. So this can be used in the field. Now that's a terrifying concept in terms of what that's going to mean for secure data transfer, the sheer volume of data we're going to end up with. It's extraordinary. It's really exciting, but it's terrifying at the same time. And the final thing that I think is very compelling about what we've heard today across all of the talks is that some vendors like DDN are addressing so many of these areas of storage that we have this possibility of an integrated support platform for a number of different kinds of production system, and I think that's really key. So finally, I think that's the end of what I have to say. I'd just like to acknowledge the members of my team. So my boss, Phil Butcher, there's me, and there are, Guy Coates is actually the leader of the team who do all of the real technical stuff, so feel free to pick, he and Peter Clapham, who does most of the iRods stuff, they are the real technical brains behind this whole thing. I'm just standing here talking about it. They are here at Supercomputing, they're over there, so you can pick their brains if you wish. John Nicholson, who's also here, is our network team leader and does all of that stuff. And I ought to acknowledge Shanthi, who runs our database team and keeps our Oracle rack stuff going for the iRods backend. And our numerous bioinformaticians who periodically break things, but also have provided us with some very useful feedback and collaborations with Renzi in terms of improving iRods. So this has been it's been quite successful. Thank you.